Hi guys, Miss Francis here. Today we're going to talk about experimental design and the scientific method. But first I just want to review how we're going to start each day or how you're going to start your day in this class. So I start out on my Google Classroom homepage. I'm going to pretend like this is Environmental Science 5-6, so I'm going to click on that. I'm going to go to the Classwork tab at the top. And then I'm going to scroll down and click on today's weekly schedule. Then I can click on the actual weekly schedule. Go to today's date. So I'm going to pretend that today is Tuesday, September 15th. And like I said, I'm going to pretend like I am in period 5-6 class. You can see that for the first period, we'd learned about our digital ecolog and what command terms were. And for the second part of our double period, we're going to be talking about experimental design and taking notes. So here you can see I can pull up the experimental design presentation, and then I can see that the experimental design notes have been assigned in Google Classroom. So if I go back to our Google Classroom, I can see that yes, in fact, Tuesday, the experimental design notes are uploaded. So I'm going to click on the notes, and then I can use this notes sheet to follow along as I am going through the presentation. So the first thing you're going to do is you're going to make a prediction. You're going to complete the chart below to discover how much you already know about experimental design and the scientific method, and you'll be shocked to realize how much you actually already know. So pause the tape here and try and make a prediction. Try and complete this table using some key vocabulary words that you need to know in order to understand how to create an experiment. So here are our notes for today. So let's jump in. You might have talked about the scientific method or experimental design. It can be referred to as both of those. It's the same process. So how does science work? Well, first of all, you need to have that curiosity driving what you want to wonder about. So there are many different facets or many different parts to how science actually works. This is the prediction that you're going to make. You're going to discover what you already know. And in fact, you already know quite a lot about experimental design and the scientific method. Now, the first step in conducting any sort of investigation is to make observations. Use your senses. Watch things, taste something, hear something. These are objective facts. You're not making any inferences. So, for example, my eyes observe the sun shining outside. That's the observation. I'm not going to make the inference that since the sun is shining, the temperature must be in the 80s. While that might be true, that's not part of making observations. And then you're going to conduct background research. You ask a question. What research has already been done on this topic? What's previously known about the topic? And then you build on that. What still needs to be known? What has already been confirmed? Then you form a hypothesis, a testable statement. It can be in the form of if, then, because. It doesn't always have to be in the form of if, then, because. And it contains a prediction. So a hypothesis is written as a statement, not a question. I think, I predict, that's not found in a hypothesis. It is a testable statement, and it has to be properly supported through evidence or rejected by that same evidence. Can't be wishy-washy on this. What's a null hypothesis? This might be something new for you guys. It predicts that there's no relationship between any two phenomena. So let's say you're testing the effect of light on plant growth. Well, the null hypothesis would be that there is no effect of light on the growth of plants. Now you're going to design your experiment. And your experiment requires a control group and an experimental group, where the control group is the baseline or the standard situation, and this group acts as the basis for comparison. On the other hand, you've got the experimental group. Independent variables, that's what will be changed. Now, I said independent variables. I misspoke. In any experiment, you can really only have one tested independent variable. Otherwise, how do I know that it's not this independent variable or this independent variable that's causing a change in the dependent variable? And the dependent variable is what will be measured. So for example, I can't test will light and fertilizer affect plant growth, because how do I know whether it is in fact the light or the fertilizer that's affecting the growth of the plant? I can test them separately as independent variables in two separate experiments, but I can't test those together. Controlled variables are also known as constants. Those are factors that remain the same regardless of the experimental group or the control group. So for example, if I'm not testing temperature, then I need to keep temperature the same. In that plant experiment that I just mentioned, I would want to keep the species of the plant the same, the environment in which that plant is kept the same, how much I water the plant the same. All right, mix dry is a clever way of remembering which axis the independent and dependent variable will appear on and what the independent and dependent variable do. So for example, the independent variable is what gets manipulated in the experiment, and if I were to graph it, it would appear on the x-axis. Whereas the dependent variable is the variable that is responsive. In other words, it responds to the independent variable, and it would appear on the y-axis. Now, I might have more than one dependent variable. I will only have one independent variable. Furthermore, dependent variables might be qualitative or quantitative. Quantitative meaning they are measured in numbers, qualitative meaning it is some sort of characteristic that's observed. So some common issues that you run into when you're designing an experiment is my sample size is too small, I didn't have a chance to repeat my trials, I had variables for which were outside of my control, I didn't have a control group, and I had more than one IV, which means independent variable. Now, the fifth step of creating your experiment is to actually do the experiment, to gather the data, to collect information from the experiment, either qualitative data or quantitative data. Once I have that data, now I can analyze it and draw conclusions. In other words, I interpret my results. Do I have any trends? Do my results support my hypothesis? So now let's go back to the predictions and let's check your answers. So constants kept close to identical as possible among all the subjects, or it won't be clear what's actually affecting the outcome. The control group is used as comparison. The controlled variables, that's the same as the constant. Dependent variables, they're measured, they respond to the independent variable. Experimental group, that's what receives those changed conditions. They receive the independent variable. A hypothesis is a specific testable prediction. It's a statement. Your independent variable, it's what's being changed, where you only have one independent variable per experiment. Large sample size increases the accuracy and the validity of the experiment. Qualitative data is descriptive, non-numerical information that I collect, whereas quantitative data is numerical information. And then lastly, repeated trials also increases accuracy and validity. So you'll see H and K can be reversed if you put those two in the opposite space. All right, so that is all you need to know about experimental design, and I hope you have a great day.